So the first presentation is Raghav Tripathi from Westview High School in Portland, Oregon.
Good afternoon, everyone. My name is Raghav Tripathi, and today I'll be presenting my work on the design and synthesis of novel fatty acid binding protein inhibitors for anti-inflammatory and analgesic effects. Now, pain is associated with a wide variety of illness and injury, whether as a symptom of a disease or in a condition in which pain itself is the primary issue, as in headaches or neuropathic pain. While acute pain, pain that is felt within the body to keep the nervous system alert for potential injury, is triggered within the body, chronic pain involves pain signals within the body firing for extended periods of time, even when someone is not feeling something painful. Over 1.5 billion people worldwide suffer from chronic pain, a detriment to worker wages and productivity, along with quality of life as a whole. Furthermore, acute pain and inflammation as a result of numerous diseases and/or accidental injuries afflicts billions worldwide. However, current anti-inflammatory and analgesic pain-killing regimens are replete with unintended side effects, causing many such agents to be removed from the pharmaceutical market entirely. Conservative estimates indicate that approximately 133,000 patients are hospitalized annually due to gastrointestinal issues from painkiller use alone, and the Food and Drug Administration estimates that painkiller use accounts for approximately 20,000 deaths annually. Additionally, incidents of abuse of opioid painkillers have risen markedly within the past few years. From 1994 to 2001, the United States saw a 117% increase in the amount of emergency room visits due to opioid abuse alone. Furthermore, extended painkiller use often leads to physical dependence and/or addiction. Now, because of all these reasons as more, it is clear that a new class of anti-inflammatory and pain-killing or analgesic medication is utterly necessary. Now, it is widely accepted that THC, the active ingredient in marijuana, has powerful effects on the central nervous system that often mimic natural molecular keys. THC is a potent painkiller that was found to lock into the cannabinoid receptor present on nerve cells. Scientists reason that since THC is not naturally present within the body, there must be a natural key molecule with a very similar shape that activates this receptor. Recently, Michel et al. Found this fabled ligand. They found a fatty acid inside the body known as anandamide, or AEA, elevated levels of which result in beneficial effects on pain, stress, and inflammation, and may further be used to ameliorate the effects of drug withdrawal as a result of addiction. Other scientists later found evidence that anandamide provides both anti-inflammatory and analgesic effects as it binds the cannabinoid receptors CB1 and CB2 present on cell surfaces. Furthermore, an increase in endogenous anandamide concentrations is hereby postulated not to have the unintended side effects associated with contemporary pain-killing medication due to its natural existence within the body. Now, since anandamide is a fatty acid, it is hydrophobic and cannot travel within a cell alone. The fatty acid binding proteins, or FABPs, are newly discovered intracellular transporters for the endocannabinoid anandamide. These FABP anandamide transporters are found intracellularly and allow anandamide to attain a temporary soluble state so that it can move freely within the cell. FABP effectively transports anandamide from the CB1 and CB2 receptors present on nerve cell surfaces to the fatty acid amino hydrolase, or FAAH, where it is hydrolyzed and digested by the cell. Now, it is clear that the inhibition of FABPs offers a novel, innovative approach to interfere with the breakdown of anandamide and corresponding increase of anandamide levels at the cell surface. The inhibition of certain specific FABPs may have advantages over FAH inhibition, since FABPs show tissue specificity in distinguished types, a property that is not necessarily present with regards to FAH. For example, in this way it will be possible to design inhibitors for neural FABPs, which are FABP5 and 7, but not those present in adipose tissue, FABP4. As such, the premise of my project was that FABP anandamide inhibition provides an innovative target for a new class of analgesic medication. Thus, the goal of my project was to design drugs to inhibit the uptake of anandamide into neurons through slowing the activity of the FABP anandamide transporter. Other neurotransmitter-affecting medications, such as Prozac, a selective serotonin reuptake inhibitor commonly used to treat depression, successfully use this approach as well. Now, I began by computationally screening compounds for FABP inhibition. After this, I purchased the best compounds and ran in vitro assays using FABP5. Then I created an optimization library for the leading FABP inhibitor and ran cell culture assays with it. Finally, I designed, synthesized, and computationally analyzed further modifications to the FABP inhibitor molecule. Overall, the aim of my project was to create a so-called painless painkiller, a new class of anti-inflammatory medication without the unintended side effects associated with contemporary pain-killing medication through increasing endogenous levels of anandamide. Now, the first phase of my research was to computationally design an FABP inhibitor. I used high-throughput virtual screening, a technique which essentially finds the binding pocket in FABP to which the inhibitor is to bind and screens millions of molecules to see which is the highest binding affinity to inhibit FABP. My computational screening employed the zinc database of commercially available compounds, which is freely available for academics and currently consists of approximately 21 million purchasable compounds. A subset of zinc was employed, containing about 1 million compounds. A virtual screen was performed using the DOC program on a supercomputer to identify potential lead compounds that could inhibit the transport of anandamide by FEP5 and 7, which are the specific FEPs implicated in this research. Out of the 1,057,042 virtually screened compounds, 48 compounds are found as hits as per the relative DOC energy scores. Then, these compounds were purchased from the ChemDiff library and assayed in vitro against FEP5 using a fluorescence displacement assay. In this assay, NBD steroid is the natural fluorophore of FEP5, which means that it binds automatically to FEP5, and the energy it produces as a result of this binding is released in the form of fluorescent light. Each of the 48 compounds was put into a solution of NBD steroid and FEP5, and as a competitive FEP inhibitor binded preferentially to FEP5, the fluorescence decreased. As such, the compounds which caused the greatest decrease in fluorescence were predicted to be the leading ligand inhibitors for FEP5. Among the hit compounds identified from the zinc database and assayed in vitro from ChemDiff, truxilic acid, or SBFI26, was predicted to be the leading ligand inhibitor for FEP5. Now, I decided to further investigate truxilic acid not only because of these results, but also because it shared the molecular core of an ancient Chinese herbal medication known as incarbilitine, once used in the ancient days to treat chronic pain and rheumatism. Assays were then done to evaluate the efficacy of SBFI26 in preventing anandamide uptake rather than only inhibiting FEPs in cell cultures. It was found that SBFI26 significantly reduced anandamide uptake in wild-type cells, giving strong support to the hypothesis. It was also found, however, that it does not significantly reduce anandamide uptake in cells bearing a knockdown of FEP5. This indicates that the beneficial effects of truxilic acid are indeed a result of FEP inhibition. To verify this further, it was also found that SBFI26 did not inhibit hydrolysis of anandamide by FAH. Now, it is clear that truxilic acid is a highly promising lead compound with significant biological activity. Because of this, I moved on to modifying and optimizing the compound for potentially increased in vivo efficacy. First, an optimization library was created around the compound. In order to ascertain the effects of modifying a specific functional group on truxilic acid, I designed a synthesis procedure for both alcohol and amine modifications. As such, a variety of functional group modifications was designed and synthesized in high yield, including 9 esters and 6 amines. After using traditional docking to study these functional group additions, chemical synthesis of the most efficient modified compounds was undertaken. Out of the 15 modified R components, 8 showed ligand efficiencies greater than that of the unmodified gamma isomer. 9 modified R components showed ligand efficiency significantly greater than that of the alpha isomer. After synthesis and testing, the IC50 of these modified compounds should be lower, and the modified drug should have greater efficacy in providing both anti-inflammatory and analgesic effects. Now, in summary, the hypothesis of this research was that the FEP and anandamide transporters are drug targets. As such, it was further hypothesized that targeting these chaperones with the use of inhibitors will both increase extracellular anandamide levels and have both anti-inflammatory and anti-nociceptive effects. This hypothesis was tested through a multifaceted, comprehensive approach. In phase 1, novel inhibitors of the FEP and anandamide transporter were identified through the use of in silico screening of large drug libraries using the DOC program. Then, both the lead compounds and more active analogs were chemically synthesized, and the most promising compounds were tested in vitro and in cell cultures. After identification of potential molecular inhibitors, they were modified through designing both amine and alcohol modifications to the original inhibitors, and their efficacies were analyzed using traditional docking methods. In this fashion, multiple truxilic acid compounds were designed and synthesized with more efficacy in FEP inhibition than any other compound known to date, and will therefore provide a new clinical outlook for the treatment of pain, stress, and drug abuse withdrawal. Now, the implications of these findings are gargantuan. The high binding affinity of compounds designed in this research corroborates the predicted binding specificity. Now, preferential binding to a specific target is key for the success of the optimized drug in the pharmaceutical industry and verifies that the designed FEP inhibitors will have less unintended side effects than FAH inhibiting molecules. Specificity also allows for the minimization of side effects. Since anandamide is the natural substrate binding to CB1 and CB2, increases in concentration will not lead to the addiction catatonia involved in derived substrates such as morphine or THC. Now, the design compounds may also mitigate addiction to a variety of substances. Drugs such as THC, which bind to the CB1 and CB2 receptors, often have more exaggerated effects than anandamide. As endogenous anandamide concentrations increase, anandamide is postulated to bind preferentially to CB1 and CB2, inhibiting binding of THC and similar compounds through competitive binding. Similar to a nicotine patch, synthesized FEP inhibitors may be applied to increase anandamide levels and decrease both physiological and mental dependence on THC and similar compounds. Using similar methods to inhibit other FEPs may also have a diversity of other therapeutic effects for other conditions beyond pain as well. Finally, all compounds designed in this research utilize a relatively simple procedure, which is critical in large-scale chemical synthesis. Now, this suggests the potential of compounds designed and synthesized in this research to be successfully integrated in the pharmaceutical market one day once a synthetic strategy is optimized. Taken together, these results strongly support the efficacy and potential of these novel small molecules developed in this investigation for FABP inhibition. In terms of future work, first, I would like to chemically synthesize and assay the optimized tricyclic acid compounds to analyze their effects on endogenous anandamide increases. Secondly, I would like to explicitly elucidate the biochemical pathway through which anandamide binds to the CB1 and CB2 receptors in order to predict potential side effects and in order to understand more of how this possible drug would work. Thirdly, I would like to ascertain whether or not the design molecule is possibly bind to other FABP types beyond 5 and 7, as well as other molecules altogether in order to investigate potential side effects. And finally, I would like to begin in vivo tests on the designed truxylate drugs. These synthesized compounds may contribute to the next generation of innovative analgesic medication and may one day be implemented on the mass scale. Thank you very much to the Siemens Foundation, College Board, and George Washington University for allowing me to present my research here. I would also like to acknowledge Stony Brook University and the Siemens Fellowship for funding my research, my mentor, Professor Iwao Ojima, my supervisor, Mr. William Berger, all family, friends, and supporters, as well as all the judges for being here today. Here are my references. Thank you.